Hello and welcome to SAM Biology class. Friends, in order to understand the structure, function and the evolutionary history of the human genome, we should know its primary structure. The primary structure of the human genome is the linear order of 3 billion nucleotide base pairs that constitute chromosomes of the human genome. The primary structure of a genome can be determined by DNA sequencing methods. Two separate methods are used for DNA sequencing. One is Maxam-Gilbert's chemical cleavage method and the other is dideoxy chain termination method. First we shall discuss about Maxam-Gilbert's chemical cleavage method, which was developed by Alan Maxam and Walter Gilbert in 1976 to 77. Maxam and Gilbert's chemical cleavage sequencing protocol uses chemical cleavage at specific base of the labeled DNA strand in order to generate a nested set of labeled DNA fragments. What do you mean by nested set? In a nested set, if we will arrange all the DNA fragments in the increasing or decreasing order of their size, then successive fragments will differ by one base at a time. In this protocol, two chemical reactions are used for cleavage of single-stranded DNA. First reaction cleaves the DNA strand at adenine and guanine, and the second reaction cleaves at thymine and cytosine. The first reaction can be modified slightly for cleavage at guanine, and similarly the second reaction can also be modified to cleave at cytosine. In both of these reactions, the cleavage of single-stranded DNA is achieved by the same process that include number 1, chemically modifying the nitrogenous base, number 2, removing the modified base, and number 3, breaking down the sugar phosphate backbone. The procedure that is used for cleavage of single-stranded DNA at guanine is like this. A methyl group is added to guanine. After this, the methylated guanine is removed by heating, following which the exposed sugar is also removed by heating in alkali. The procedure that is used for cleavage at adenine and guanine is identical to the procedure that is used for cleavage at guanine, except for the fact that an dilute acid is added after methylation. For cleavage of the single-stranded DNA at thymine and cytosine, hydrazine and piperidine are used. In sequencing, the first step is to generate a large number of copies of the DNA strand. This is achieved by cloning. Single-stranded DNA can be cloned by using M13 phase as the vector. Similarly, double-stranded DNA can also be cloned in suitable vectors. In case of double-stranded DNA, after cloning, the two strands are separated, then they are sequenced separately. After cloning, all these fragments must be radiolabeled at the 5' end, for example by using phosphorus 32. Radiolabeling is necessary because the radiolabeled fragments can be seen in autoradiogram. From this autoradiogram, we can identify the position of different DNA bands from which we can determine the sequence of nitrogenous base in the DNA strand. For example, we want to determine the sequence of this double-stranded DNA. After obtaining a large number of copies by cloning, all such copies are radiolabeled at the 5' end by using phosphorus 32. After radiolabeling, the two strands are separated, then they are sequenced separately. Now we shall see as how the single-stranded DNA is sequenced by the chemical cleavage method. All copies of this labeled single strand are divided into 4 different batches, which are subjected to 4 different chemical reactions. These 4 different chemical reactions will generate 4 sets of labeled and unlabeled fragments. In first batch, cleavage will take place at guanine, and in second batch at guanine and adenine. Whereas in third batch, cleavage will take place at thymine and cytosine, and in fourth batch at cytosine only. All these 4 different chemical reactions will generate 4 sets of fragments that will include both labeled and unlabeled. In first batch, cleavage takes place at guanine. When the first guanine is cleaved, we will get 2 fragments. The left side fragment is radiolabeled and the right side is unlabeled. Similarly, when the second guanine is cleaved, we will also get 2 fragments, both labeled and unlabeled. Similar fragments will also be obtained when the last guanine is cleaved. In case of second batch, due to the cleavage at guanine and adenine, we will get more fragments. First 3 fragments are obtained due to cleavage at guanine, and the second sets of fragments are obtained due to cleavage at adenine. In batch 3, cleavage takes place at thymine and cytosine. As a result of this cleavage, several fragments are generated, out of which these fragments are generated due to cleavage at cytosine, and remaining fragments are generated due to cleavage of thymine. In this set of fragments also, the left side fragments are all radiolabeled and the right side fragments are unlabeled. Finally, in batch 4, cleavage at cytosine is 3 labeled and 2 unlabeled fragments. After chemical cleavage, all such fragments that are obtained from 4 different chemical reactions are separated by polyacrylamide gel electrophoresis in 4 separate lanes. Patch or polyacrylamide gel electrophoresis is a powerful technique which separates charged biomolecules based on their molecular size. By this technique, even the DNA strands which differ by one base only can also be separated. When subjected to electrophoresis, all these labeled and unlabeled DNA fragments, which are of different sizes obtained from 4 different chemical reactions, will migrate at different rate in the electric field. As a result, they will be separated into different bands. Only the bands containing the labeled DNA fragments will be visualized in the gel by autoradiography. Take the example of labeled fragments that are obtained from batch 1 chemical reactions. From batch 1, we will get only 3 types of labeled fragments, out of which this is the shortest fragment. As a result, this fragment will migrate at a faster rate and it will be separated into a band which will be nearer to the bottom of the gel. This will be followed by next shortest fragment which will move comparatively at a slower rate, and it will form another band which is located above the first band. And finally, the next shortest fragment will migrate even at a slower rate, and it will also be separated into another band which is located above the second band. Similarly, the other fragments that are obtained from batch 2, batch 3 and batch 4 chemical reactions will also be separated into different bands based on their molecular size. As already mentioned, after electrophoresis, all these DNA bands can be visualized in the gel only by autoradiography. After autoradiography, we have to read the gel from bottom to top in 5' to 3' direction. In order to know the sequence of the DNA strands, start with the lowest band which has been generated due to cleavage of thymine. So the first base will be thymine. This will be followed by second band which has been created due to the cleavage of guanine. So the next base will be guanine. After second band, move to the third band which has been generated due to the cleavage of adenine. So the third base will be adenine. This third band will be followed by fourth band which has been generated due to cleavage of cytosine. So the next base will be cytosine. We have to read the gel in such a manner by comparing the relative position of different bands from bottom to top in 5' to 3' direction, as a result of which it will be possible to determine the sequence of the single-stranded DNA. One important point is that when the DNA strand is cleaved at first base, it will not show up in the gel. As a result, it will not be possible to identify the first base of the DNA strand. So we have to proceed from second base onward. So friends, this is all about DNA sequencing by Maxam and Gilbert's chemical cleavage method. For more updates, please subscribe my YouTube channel, Sam Biology Class.